lectures on neuroscience.
Okay, thank you very much, Hilda.
Jordi.
Good morning, everybody. Good morning. Yeah, let's start. Everybody's around. So today I will present 3 different topics, basically. One is that I will introduce some aspects that you should know on data analysis. So they will be important for you to take care of your assignments, okay? And I think it's actually a lot of fun to play with data. Then I will introduce some basic concepts of numerical simulations that also will later explored by Jesus. And finally, I will start discussion about machine learning in relation to neuronal cultures, and we will continue on Friday about this topic. Yeah. So first of all, I want to introduce a little bit the scary world of data analysis, because most neuroscience today, about 80% of neuroscience, deal with analyzing data that you get from something, normally experiments with humans or animals. So, and that is a dangerous topic because sometimes people don't know exactly what it's doing. And I want just to think about that and discuss with you this afternoon and next days, okay? So basically, just to make clear, we have to define 3 concepts. One is the structural connectivity, which is the actual layout of connections in your system. That can be at the microscopic scale, like the C elegans that we discussed the other day, okay, where you have the exact synaptic connections between neurons, or it can be at a macro scale in the human macroconnectome, in which basically you look at the connectivity between brain regions, not exactly neurons, but brain regions, okay? And something that is very beautiful that people found extremely fascinating, and is that some traits that you observed at the micro scale in the C elegans, for instance, particularly modularity, is repeated. This concept of modularity is repeated across different species along development, from the C elegans to the macro scale of the human connectome, okay? So that's why complex networks are so amazing for all of us, because you have a series of features that are present in different neuronal systems, okay? And I want you to have this in mind, okay? And then, in general, we define two ways of looking at the interaction between neurons. One is the functional connectivity, in which I look at the statistical relationships between firing neurons or brain areas. And basically, I get undirected and typically unweighted graphs, but they have to be undirected, because with this statistical relationship, I cannot get information about the directionality of the interaction. So basically here, what I look typically is at 2 signals, and I ask myself if they are similar or not. And then the third one is the effective connectivity. In this case, I have to use causality concepts, okay, to infer the directionality of the interaction, like here. Okay? These 2 neurons, 5 and 2, are effectively connected because the activity of neuron 5 causally affects the activity of neuron 2. And today, Oswaldo will discuss more about this topic. But to put an example, relatively simple, okay? I will discuss in a moment what we do, for instance, in the context of transfer entropy, in the context of cross-correlation, which is the material that you can play with the data I gave you yesterday. And here is, to conclude this part, here is just a summary of the current efforts in the community. So if we consider the brain as our system of interest, then on the one hand, we can extract the structural connectivity somehow. That's, of course, very difficult, but we get the actual connections between brain areas. And on the other, using different techniques like fMRI, EEG, or others, we get the functional connectivity. And of course, the goal is to see how they are related, particularly in disease. And we will go to that later. But conceptually, this is what people is exploring in different fields. Yeah. So yeah. So before analyzing anything, so you have to be careful with that. Okay. So you have data, and you want to play with the data and get something, but you have to be careful. First of all, you have to know if there are artifacts or not in your data. That is very important, because you may get some results that look very, very cool, but maybe they are related because, for instance, in EEG, people is breathing and their heart is beating. So there are artifacts there that you have to be aware. Yeah. Also for the fMRI, for instance, in neurons, if I change, this is data from fluorescence calcium imaging. Remember, this is the characteristic experiment that I have with me. Oops. Okay, this is a typical experiment of calcium imaging, but the interesting thing is that this events over there, these events that you have here, okay, correspond to the rise of the fluorescence signal when my neuron is firing. But if I change from this fluorescence probe that is called GCAM6, if I change to another one, okay, this profile may be different, yeah? So I have to be careful with that. The same kind of, let's say, the physical system, let's say, but the kind of data I can get can be different. So one has to be careful, okay? Also, I think for me it's very important that you know, as best as you can, the analysis tools that you are going to use. So I don't want to offend anyone, but I had bad feelings, bad relationships with the medical community and sometimes with biologists because they want you to just, no, I don't want to look at you doing, I want to close the eyes, press the button, and get the results. And normally the ones I like. No, you have to be careful on that, and you have to try to make an effort to avoid that, okay? To avoid that. And you have to really try to understand as best as you can the methods that you're going to use, yeah? And then for that reason, you can use simple models, numerical simulations to play with artificial data and the tools that you are going to use, and that gives you an impression if you are going in the which direction or not. Yeah, and of course you have to enjoy it. You have to enjoy it. You have to invest time and invent data by yourself, add artifacts, add noise, whatever you like. But at least you will get a feeling of what's going on there. So please, this is the basic idea, okay? And then let's put an example so you understand what you can do with the data I provided you yesterday. And I decided to use cross-correlation because it's very intuitive, right? Everybody, I think, knows cross-correlation. Everybody knows it, right? Or who doesn't know it? Okay, it's familiar, okay. So conceptually, you're multiplying two signals. So if they look very similar, cross-correlation is close to 1. If they are very different, it's close to 0, yeah? Conceptually is that. And you can also shift, if you want, a little bit the two signals. So, and to look for the maximum of the cross-correlation. And you can use that information to get a feeling of the possible directionality of the interaction. Okay? But at the moment, let's make it super simple. I take two signals, I multiply them, cross-correlation, and I see how they look like to one another. Okay? So cross-correlation is very famous, is used by many, many people. And normally you get something like this. It's of course, a symmetric network because it's an undirected graph. And then the closer you are to one, the more similar are the signals that you were exploring. And of course, this is a typical result from the literature. And of course, what is really fantastic is when you can compare your functional interactions, I mean the communication or the interaction among neurons with the structural connectivity. This is something that people is really interested because, in particular in the context of medicine, you want to see whether damage, structural damage, due to the death of neurons, for instance, or the loss of connections, has an implication, is somehow related to some changes at a functional level, okay? So, but this is a dream, yeah? So normally you do not have access to the structural connectivity in parallel to the functional one. So this is something that you have to be aware, yeah? Okay, good, very good. So actually, any of you know this? Who knows this? Oh, okay, this example. Only one person? Also two, okay, few people, okay. Three, okay, very good. So this is a joke somehow, but it's very illustrative about the problems of analyzing data, okay? So one guy in UK bought an fMRI machine. He paid €1 million for that. And he said, okay, I'm going to put people here. Let me check what's going on with this machine. So he bought salmons, put the salmons inside the machine, and then the salmons were looking at female photos of salmons, okay? And then he was looking at the activity in the brain of the salmon and wanted to see what was happening, okay, with that data. And everything looked very cool until he realized what he realized. Until he noticed that because the salmon was very dead, okay, this data was very unreliable, okay? So it's true. He put dead salmons inside the machine. And then he was of course, very pissed off, yeah? Because that shouldn't be possible, okay? So basically, the machine is detecting some noise from somewhere and is ascribing that noise as interesting behavior of the salmon. What happened? So that the scientist, didn't have access to the code, to the mathematics, to the MATLAB, whatever you want to tell it, on the analysis of this data, okay? So he was pressing the button and he was getting the results. That's why he got very angry. So he published a paper about the possible errors. Actually, the paper looks very, 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 very funny. I mean, he really, and the journal is also very cool, okay? But he published this paper announcing to the community that one has to be extremely careful, that sometimes we play with the data and we don't know exactly what's going on. And then after that, this was in 2008, And then you see a lot of papers started to appear that were serious papers describing the limits of data analysis, at least in the context of functional magnetic resonance imaging. And people got extremely angry, and now the people estimated that around 100,000 papers are wrong because people blindly accepted the results from the machine. So that's very dangerous. So this was a lot. Of course, the press made a lot of fun of that. And well, this is in Spanish, but yeah, Chinese scientists said that, for instance, the heavy metal was bad for your brain. But then when you were looking at the data again, okay, and analyzing it with different tools, you were realizing that that was not true. The problem were the Chinese scientists, not, not the data, okay? Anyway, so basically, of course, people made a lot of jokes. You can find a lot of jokes about the danger of cross-correlation, that things seem to correlate very easy. This is an example, okay? Size of ice cream. This is the green one, correlates very well with forest fires, okay? And of course, they have nothing to do with one another. Is the heat that drives them together, okay? So please be careful and take seriously time series analysis, as we discussed with Christina, and explore, of course, always different methods for a given problem that you have at hand, okay? Anyway, so then in the context of your data, so I have my data. I get the functional connectivity matrix based on cross-correlation, okay? The data is the same that I gave you yesterday. And you get this cool matrix, and this is the distribution of cross-correlation values that I'm getting, okay? And the question is, okay, I'm happy enough with these results, or should I be careful somehow, okay? Well, yeah, you have to be careful, because you have to decide whether these results are significant or not compared to something, because I don't have the structural connectivity, I don't have any reference, I have to be extremely careful. And how do I do that? I build surrogates, okay? I try to randomize my data and see whether this, for instance, these extremely low values of Pearson correlation can occur by chance, okay? That is the minimum thing that you have to do to start realizing or to start exploring whether I can believe my results or not. And this is super interesting. And surrogates, basically, what you do is you randomize your data, okay? So imagine, for instance, that I take this signal, yeah? And this is fun because you have to think on how do you build surrogates. So you have this data, for instance, and one option for building random signals is that I take a random signal with the same amplitude as this one. This is like my noise, okay, my experimental noise, yeah? And then I look at the cross-correlation, and I see this is my distribution of values. So basically, I would say that these values in blue that correspond to my original experiment, these values in blue that overlap the gray ones may occur by chance. So I cannot trust them. You follow the idea? Yeah? So one has to be very, very careful. However, is this a good surrogate or not? It's not. It's not a good surrogate. Why? Because I'm destroying the biophysics of the problem. Why? Because I have a sharp increase of the signal and a slow decay, and I don't have this kind of crazy noise, yeah? So if I want to do more interesting surrogates, what I have to do is to try to preserve as much as possible the biophysics of my problem. For instance, how do I do that? What I can do is to take this data and then take chunks of data here and swap them randomly. So I'm breaking the correlations of my system, but I preserve the biophysics of the data. The signal looks like this, okay? And it's much better as a surrogate. And then my result is that one. And you see it extends to larger values. Yeah? But this surrogate is much more interesting than the previous one. So finally, I get something like this. And of course, now I have to decide, okay, where do I cut? Yeah? So I could cut maybe here or just here where the distributions cross. That depends. You have to be careful. Yeah? But for instance, I have put here an example. So I can take, this is the surrogate that I like. This is the mean position. I can take 1 standard deviation, 2 standard deviations, 3, and so on, okay? So for instance, if I cut at 2 standard deviations, I get this one. If I cut at the maximum, yeah, of the surrogate, I get this matrix. The good thing is that I can take this matrix because I can trust these results, okay? I can trust them. They cannot occur by chance, yeah? And I can use this data to later on go ahead and get conclusions. Yeah? So please be careful. You have to understand that you have to always build surrogates to play with your data. That's it. Any questions for this part so far? No? Ah, sorry. Yes.
It might be a stupid question, but
Yeah.
can you generate a random surrogate that will you need to cut the higher correlations, because it seems quite convenient that you are only cutting off the lower correlations?
Yeah, you mean to generate new surrogates?
Yeah,
no?
that will cut off the higher correlations, because you are only keeping the super
high.
strong
Yes,
events.
but then no, but the meaning of surrogates is that you want to identify
You really want
which
to
is
have just the higher.
minimum, yeah, which is the minimum correlation that you can believe that cannot occur by accident. Okay. This is the concept of the, yeah. I mean, of course, you can invent data that is ultra correlated, but you're inventing it. I mean, you could go here, yeah, but it's an artifact. It's a totally artificial signal, yeah. But the concept is, yeah, I have my data. I want to see which is the minimum cross correlation value, from which upwards I can trust my results, that they cannot occur by chance, yeah. Okay, so this is the main result, and also in the context of what Oswaldo will explain today, you can, of course, always compare the cross correlation results with other tools, like transfer entropy with a causal inference method, and nonlinear. And basically, conceptually, you get also the same kind of modular structure and some traits that are reproducible or that are present in both. So in that sense, you could say that, okay, I'm going to start to believe my data and can start getting information, for instance, about the properties of this complex network. Okay, okay, good. So this was the first part. The second part is that, yeah, we want to do numerical simulations. Why? First, for the context of the previous experiment, I can simulate neurons. I can put calcium imaging, let's say dynamics. And from that, I can get data that is very similar to what I have experimentally, and that allows me to keep playing and exploring and try to see if the behavior, that the experimental behavior I'm seeing in my lab, I can understand it in the context of an in silico modeling. So this is something very cool. All of us, yeah, most of the people that is doing experimental neuroscience always tries to go to the computer and try to see if I can reproduce qualitatively the result I'm seeing there, okay, to better understand the basic concepts, the basic ingredients of my system, okay, to exhibit the collective behavior or other properties that I want to look at, okay? And here I put a basic summary of what we discussed the previous days. So we have a neuron as an excitable system. The neuron has to increase the voltage to fire. If I do nothing, okay, see, if this part doesn't exist, the neuron will go back to the resting state, and I need inputs. I need, for a given synapse as this one, I need a train of inputs, okay, in a short time window to increase the voltage, reach a threshold, and the neuron to fire. Of course, there are different synapses, so I have to also add this thing, okay, all possible synapses in my system and all possible trains. And of course, I also have noise, yeah? Noise that is reflected here, in which some of these spec trains also contain the black dots that correspond to random activations in my synapse, okay? And also remember that in general, we have excitatory and inhibitory inputs, like this represented here. The inhibitory inputs, they decrease the potential, the excitatory ones increase it. And conceptually, this is something that it's important for us. One has to achieve this condition that the number of excitatory and inhibitory inputs have to be, in a small time window, has to exceed a threshold for the neuron to fire. And this is called quorum, and I think that Jesus will also use it later. So this is an important thing. Also, and I introduced the different models that we can use for a neuron. We have the Huxley from 1952, then the Fitz-Unagumo simplification from the 1960s that takes basically, this is the node that Anna discussed yesterday. And the point is that people realized when looking at this plot, and is that actually most of the interesting behavior of a neuron happens in this region, okay? Yeah. So people thought, okay, why do you have such a complicated nucline with this cubic nonlinearity? But maybe you can just make something like a parabola here, okay? Like a parabola here, and make it quadratic. So this was the concept that Mr. Izikevich developed in 2007, in which we realized that actually 90% of the neurons, if you make this simplification here, like this parabola, these 90% of neurons behave very, very, very well. So you don't really need sometimes to used the original Fitsunogumo or the Honshin-Huxley. You have to be, of course, please be careful. This doesn't mean that the other models are useless, no way, okay? But if you have experiments, or, no, in this case simulation, sorry, if you have simulations in which you are super interested in how complex behavior emerges, for instance, because you are exploring different networks, then this model is very, very good because you're giving more importance to the phenomenology associated to the connectivity of the neurons rather than their intrinsic dynamics, okay? So there is something very important. And as you know, I also work in medicine. So for instance, if I want to explore problems related to medicine, maybe it's important for me to take into account aspects such as ion channels that don't work properly, okay, or the neurons that have some genetic problem and they don't fire as they should, okay? So when this happens, I have to go back and use the original models, correct? Yeah. So at the moment, for simplification, the Izikevich model works very well, okay? Questions so far? No? Okay, so the Izikevich model is then reduced to these equations with this quadratic term here, and conceptually, what Izikevich did was, okay, I care about this membrane potential, and I could use some kind of recovery variable that once the neuron has fired, the neuron fires, and once the neuron has fired, this recovery variable takes action and goes back the neuron to the original resting state. And what Izikevich did was to use some kind of threshold condition, very simple threshold condition, and is that as soon as the neuron has fired, has elicited an action potential, I reset the system so it goes back to the original state. And of course, you also have noise, of course. So basically, conceptually, when you want to simulate this system, what you do is that if you reach sufficient events, okay, in a shortened window for quorum to be achieved, then the neuron fire, you have these dynamics, and it returns to the original state. Something that is also, sorry, yeah. Oh, sorry, I cannot. This is Izikevich.
So you are approximating as a parabola. So this is a harmonic oscillator, or is some other approximation with quadratic function?
Oh, sorry. Maybe you can just,
it's a harmonic oscillator because you're approximating as a parabola. So
if it is a neuron, sorry.
Harmonic oscillator.
Sorry. An harmonic.
Yeah, because it's a parabola. So I thought maybe.
No, but it's but harmonic. Maybe we can discuss it later. Okay. But sorry, yeah, maybe it's easier. Yeah, sorry, thank you. And okay, so the point is that in addition of these equations, okay, for the model, something that is also funny to introduce very quickly is the fact that the synapses get tired. Okay, conceptually, there is something called short depression, and what happens is that for each time that the neuron has fired, we have this. This is the weight, the strength of the input of the synapse. So this quantity d, okay, it gets reduced by a factor beta, with beta is lower than 1 each time that the neuron has fired. What I mean is that d at the beginning is 1, then the neuron fires and d goes to 0.9, then to 0.8, to 0.7, and so on. Keeps decreasing. So this means that the capacity for a neuron to fire a lot in a super fast, it goes down. Okay, so practically after few events, the neuron is totally tired and cannot fire anymore. And this is conceptually, this is related to the fact that you have neurotransmitters that move from one side of the presynaptic terminal to the postsynaptic terminal, and these neurotransmitters, they cannot produce instantly. Okay, biologically, you need time to make them. So this fact of depression quantifies this exhaustion of the neuron, okay? And this is very important because this means that when you simulate a neuronal network, you see that in reality, neurons can fire a lot, and then they have to rest. They have to rest for a while, and then start again firing. Okay, and these are the ingredients, and finally, to complete this part, is also something that I discussed during the hang-ons yesterday. You also have to take into account that neurons are not embedded, let's say, in a mathematical, let's say, topological entity without metrics, but neurons are actually growing either in the brain, in a surface, or in a structure that combines two-dimensional and three-dimensional organization. Remember that we have the brain and we have the cortical columns, and the cortical columns are basically 3D. Okay, so you have 2D and 3D, but in the context of the neuronal cultures, if you play the neurons on a glass cover slip, basically they will connect, of course, following the surface that you have, and typically neurons will connect to the neighborhood, and long-range collections are rare, okay? And you can explore this a lot, you can have a lot of fun. For instance, you can put obstacles in your system, so the neuron is growing, the axon is growing, touch an obstacle, gets deflected, and you can investigate this a lot and have a lot of fun, okay? But conceptually, the super basic thing is that you can take the neurons on this metric space, and then, for instance, say that those neurons that are in a neighborhood of my original neuron will connect with higher probability than others that are very far away, okay? That is a basic concept. And then, as I said, if you want to make it much more funny, much more interesting, you can introduce obstacles. For instance, here in this illustration, I have triangles, and you see the neurons that are there. The axons will have an easy access, okay, to these angles here, and it's very easy for the axon to keep going down, okay? But for an axon that starts growing in this part, it will be very difficult to find this tip, okay? So it will be highly improbable that goes up, okay? So conceptually, by using these kind of traps, I can funnel, okay, the connectivity of my system. And actually, this is fun because experimentally, I can also do the same, yeah? So numerically and experimentally, I can match different systems, okay? Okay, and finally, you plug everything in the computer. For instance, here I have a network that is described in this context, okay? Nearby neurons tend to connect with higher probability than distant ones. You have something like this, okay? And then you simulate the network in your computer. Of course, you have to introduce noise, of course, yeah, which is all these things that you see here. And from time to time, you get these bursting events that are very similar to what we saw in the experiments, okay? So it works, and then you can do a lot of fun. This is a typical experiment that I have. So basically, yeah, you can play with your simulations, and your experimental data and compare one another and try to understand which are the ingredients that shape this kind of bursting behavior in the experiments, okay? Note that I think I told you the other day, the fact that neurons basically connect randomly, one second, the fact that neurons basically connect randomly with the others facilitates this kind of bursting regime that you can also see in these simulations, okay? So conceptually, at least with these very basic simulations that I'm presenting here, I'm able to reproduce the very basic behavior of my neuronal culture in the laboratory, okay? And with that, I'm basically very happy. Oh, sorry, I'm here. No? Whatever. Well, sorry, yes, go ahead.
I'm sorry if I'm asking something that you presented in your first talk. I couldn't
Don't
see.
worry, yes.
But you're showing experimental results and simulations, right? These videos are experimental results, or
this one?
You showed some of the experimental results, right?
Yeah.
Something that I just curious is how many types of neurons you have in your culture? I suppose it's just one, and what kind of synapses they make? Are they excitatory and inhibitory,
yes,
or
exactly.
both?
Yeah, here we have typically in these cultures. Oh, the computer. One moment, the computer hung up. One moment.
Okay.
I did nothing. The computer just got a bit crazy. Maybe we can ask Tiago, maybe. Or so yeah. While we fix this thing, yeah. In the cultures that we have there, we have 80% excitation and 20% inhibition in the system. Okay, and what we know from immunostaining and other techniques, and is that typically neurons connect to the neighborhood. Okay, so if a typical culture in our lab is 3 millimeters in diameter, the typical length of the axon is about 1 millimeter. It's very rare that you have a very, very long axon crossing the entire culture, okay?
Just for me to understand the experiment, you put the two types of neurons, the excitatory and inhibitory, and they grow in In proportion
Exactly.
to the number of put initially.
Exactly. So basically, yeah, so yeah, so basically, you're totally right. In this case, for this particular example, okay, the neurons that we have there come from primary cultures from rat. So basically, you dissociate the tissue, and then you plate the neurons there, and basically you get a homogeneous soup, yeah, of excitation and inhibition in your network. Okay, this is okay. Let's say that this is excitation and this is inhibition, and in total you have 80% of excitation. Yeah, and about 20% of inhibition.
Excellent.
Okay, thank you. But something that people asked the other day, which is now good to discuss again quickly while we fix the computer, and is that there is no way at the moment with primary cultures to tell if excitation has to go there and inhibition has to go there, or there has to be long range or short range. It's not possible. But people in our lab and other people and other groups were trying to develop tools using induced pluripotent stem cells to prepare excitatory neurons and inhibitory neurons separately and then try to connect them as we wish. But that's very, very difficult, okay? Yeah, so thank you for fixing the computer. Yeah, so this is the example I was providing before, okay? So yes, so this is 80% excitation, 20% inhibition, and this is what you get, okay? This bursting behavior. Of course, you also simulate that in the computer, and the funny thing, the good thing is that I could make predictions. So in the computer, I could shut down the inhibitory neurons, or the other way around, shut down excitation, and see what's going on in the computer, and then go to my cultures and see if I can see the same behavior. So that helps me understanding what happens at the collective level, okay, when I modify the connectivity of my network, okay? Yeah? And then
how do you, it's me, how do you simulate the traveling waves in your computer? I didn't see any traveling on the left, I'm sorry.
The simulation is this one.
Sorry?
This is the simulation that replicates, the simulation that replicates the behavior that I see experimentally, in the sense that 80% excitation, 20% inhibition, and you see I naturally get this bursting behavior. Yeah?
Or that you saw some excitations traveling around.
No. No. That would be nice. I mean, to control excitation, yeah. But conceptually, conceptually, this is what we have. And of course, I invite you to keep playing with the simulations, okay, to go as far as you can go. And something interesting that with Jesus we explored. Okay. ¿Qué ha pasado? No lo sabemos, ¿no? Vale. Pero está compartiendo todo el rato, ¿no? Vale, mala suerte. Está aquí, gracias. Vale, es esto, vale, luego lo arreglo. Se cuelga por razones que no entendemos, ¿no? Vale. But the computer is all the time sharing, right? So I have to share again, okay. Okay, I think that for some reason. We don't play the video. Yeah, I will not play the movie anymore. Okay, so if you want to see the movie again, please wait later. Okay, good. So this is okay now. So the last thing I want to show related to this part is that something super cool that you can investigate is whether activity always starts in the same place or not. I mean, if I take this data, okay, I take this data and you see that it's bursting, yeah. But if I look at this data in detail, actually what I see is that activity is a fast propagating wave, yeah. And that's super, super interesting. To achieve this, what I have to do is to record the experiments at very high speed, yeah, at 100, 200 frames per second, super fast. And then what I see is that activity is a fast propagating wave. And if you notice, activity starts more or less always in the same place, more or less here, okay? So this means that something happens at a dynamical level that favors, at the dynamical together with a network that favors initiation in the same spot, okay? So what you can do? You can do experiments, for instance, investigate where these spots concentrate, how they change in time, okay? You can do a few things experimentally, and of course, numerically, you can explore why this is happening. And if you are curious, you can take a look to some of all these papers, that these papers discuss how can you understand that initiation occurs in the same place. And something fascinating that I explored with Jesus, and is that if I look at the statistics, okay, of this initiation of activity, yeah, if I look at the statistics, and then in the computer, I kill the neurons that initiate activity, what I realize is that activity starts somewhere else, and somewhere else, and somewhere else, and so on. And that's super, super cool. And then you can see experimentally that the same happens. So basically, the neuronal cultures are extremely resilient to damage. Okay, that is something that we found extremely fascinating for us. And yeah, if you want more details, you can look at the papers. You can discuss with me later. And Jesus will also, in his talk, provide a bit more of information. But it is super, super interesting because you're using the simulations to make predictions of your experimental behavior. Then you go to the lab and you explore what's going on. Okay. Anyway, more questions of this part so far? No? Then I will start. We'll change topic at the moment, or a little bit, and I want to start a bit of discussion that we will continue next day. And it's about the concept of artificial intelligence, because I'm trying to deal with that in my lab as well. And of course, it's something that is now very controversial with many things going on. Yeah. So I want to take a look at this. Yeah. So artificial intelligence, of course, all of you know it, and I think some of you are also working on it. But conceptually, both artificial intelligence and machine learning are brain-inspired concepts, actually computational resources, for the automatic decision-making and data classification. Okay, this is what we do typically, many of us, when we buy a package from some company, from MATLAB, okay, to try to classify our data. But it is brain-inspired, and we will explain what that means. Well, it means that basically we use simple, or relatively simple, neural networks in which we have some kind of ideal neurons that they are connected to other neurons. So this is our input layer, the output layer, and we have some kind of hidden layer that can be very, very big, in which I tune the connectivity of my neurons. So given an input, I have an output. And if I train the system with a lot of data, I can get a system that is able to classify my data or even make predictions where when the input signal is not exactly what I was expecting. Okay, so we can play a lot with it. And conceptually, artificial intelligence, machine learning are related to this concept of neural networks. And people like to visualize it like this Russian doll over there. So neural networks is the first one, and then you have machine learning and artificial intelligence. So conceptually, machine learning represents the capacity of a system to learn and improve from experience. And basically what you do is you program algorithms in the system that so it can analyze the data and find interesting patterns, and then I'll learn again and again based on this data. However, artificial intelligence is much more advanced, and basically you try to mimic cognitive functions that are typical from human intelligence, like natural communication, understanding of the world, let's say of our environment, or even to provide assistance to other people. Yeah. So basically notice that they are very different, and now with ChatGPT we are in this region here in which we really want to have human-like machines, okay, that help in our daily life. And the problem is that, well, yeah, the problem is that this is extremely expensive, extremely expensive from a computational point of view, okay? And here, just although I know that everybody is familiar with these concepts, I just wanted to put a simple video, okay? So you realize that, of course, you take your neural network and you can train your system to do
memory is a kind of natural behavior of networks of neurons.
Okay, so this is one of the many videos I could find on the internet in which you have in the input, the activity goes, the flow of activity goes to particular locations in my network, and you keep repeating this again and again, and finally the system converges towards a particular pattern of activation for a given input, yeah? And this, for instance, the system here is able to recognize a number, okay? But conceptually, it's basically to take a gigantic neural network, train it to recognize a particular pattern. And the amazing thing is that this works, of course, this works super well, yeah? This works very well, and of course, ChatGPT appeared recently, let's say a few months ago, and since then, many friends, now I think there are around 14 different strategies similar to ChatGPT. So these friends or colleagues are really flooding, yeah, the technology world, providing solutions similar to this one. The problem is that this is a horrible nightmare in terms of computation and resources. And actually, I insist, is brain-inspired. We are not learning anything about the brain, or we'll discuss that later, okay? But we are not getting information on how the brain works by using this technology. But it's cool, it works, yeah? However, I put here some numbers that now actually the present version of ChatGPT is using from 80 to 100 billion neurons, okay? So it's basically, actually, it's like a human brain in a gigantic set of computers. So 80 to 100 billion neurons and around 100 trillion synapses, but this is everything done by software, or most of it by software and super expensive GPUs that are taking the task, okay, of simulating these neural networks. And it's totally crazy, and people realize that typically we need the energy and the water of at least 1 million people cities, okay? And now every year, or no, every year, no, every few months, we have another company that is developing the same thing, and again, and again. So it's getting totally wild, yeah? While, as Christina said the other day, the brain is working at 12 watts only, but here you need terabytes of power, okay, to feed these companies. And something that it's super interesting to me is that, okay, these systems work. I'm in favor of using machine learning, artificial intelligence, because we can really need it. And actually, I read the other day in the news that people was very worried because now humanity is producing such amount of data, such a massive amount of data, that we need artificial intelligence to digest that data and give us the main conclusions of that data. We are not able anymore to process such amount of volume of information produced by humanity. So we need these systems, okay? The problem is that the amount of resources needed are totally crazy, okay? And to keep discussing these aspects and the next day, I want to make the difference that, again, okay, that this is brain-inspired and not brain-like. And this is super cool because this allows us to develop maybe new technologies in which we can carry out artificial intelligence in brain-like systems like in a neural culture. And actually, there is a lot of interest in the community to favor research in this direction. So, and that's why, well, I will skip this for a moment, and that's why I put this example here about the visual cortex, because we still don't understand how information is encoded. So you have to imagine that I have a microscope, here I have a camera, in the visual cortex of the rat. I can look at different neurons, okay? And then I put different patterns to the animal, stripes, okay? Vertical, inclined, horizontal, and so on. And then look at which neurons are firing, okay? So for instance, here you see that neuron 19, neuron 6, and so on, okay? So different neurons are firing in response to different orientations. And that's very cool, but this data is put together and then sent, put together in the visual cortex, processed, and sent to other parts of the brain to make sense of the world, okay? But the point is that we still don't understand how the spike trains are associated with a particular image that you are seeing there, okay? But it works all the time. And something I found really cool in a recent paper from this year, actually was published few months ago, and is that not only the system is encoding information, yeah, depending on the kind of shape that you have, but there is adaptation, there is attention. So if you're looking at something and your attention is different, yeah, from the day before, you may interpret the data in a different way. And also there is prediction. So sometimes when you are seeing something, your brain is trying to predict what will happen with that image. And this makes things super complicated because the coding that you have for a particular signal also encodes for the possible prediction that you will have in the future, and also it also depends on your learning, okay, or your, let's say, education from the last, let's say, 20 years, okay. If you learn to look these kind of images, maybe your brain will process them in a different way than people that is not familiar with this kind of images, whatever, okay? So this is just to show you an example of how difficult it is to code information and understand the brain code, okay? And that's why I put here an example, and is that we are not computers, okay? Humans are not computers. And I think Anna will agree with me. I think that we process information in a way that we still don't understand how it works. But for instance, I put this example here for two reasons. One is that I participated in an exercise about 20 years ago. Oh, wow, yeah, 20 years ago. Okay. So in this exercise, I had to memorize this painting from Van Gogh. Then I had to give the promise that I would never look at this painting again, at least intentionally. And then the idea is that in a logarithmic scale, people was asking me, in a logarithmic scale, what I remembered from this painting. Yeah, so it was, so for instance, I look at the painting, and then a few seconds, few minutes, hours, days, months, 5 years, 10 years, and so on, okay? And the point is that when people was asking me questions about this painting, at some moment, I was forgetting things, okay? And I was inventing data. I'm inventing objects in the room. Basically, because based of my experience along these years, I was totally changing my interpretation or my feelings about these paintings, yeah? And for instance, I added a clock, because for me it was totally natural to have a clock in a bedroom, okay? I was adding maybe some flowers. So it totally depended on my, let's say, experience, my years of experience living, okay? So that I was changing this painting. So here in this room, we have people from different parts of the world. So for instance, if you do the same exercise, maybe in South America, I think we have someone from Africa, right? So maybe you will invent new things associated to your culture, yeah? So this means, but this is funny. So for 20 years, I was modifying this painting. But then if I go to Amsterdam and I look at this museum, at the museum where this painting is, okay, as soon as I see the painting, my brain will make the click, is that painting, you know? So I store things that made no sense related to this painting, but finally when I see it again, something happens and I can see it again. I can realize that is that painting, yeah? So we do not code information as in a computer, that we have the pixels of that data. We don't do that. We save in the brain the concept, and that concept is very difficult to understand how the hell it's coded, okay? This is interesting. And then I put also, as a joke, the 20 euro bill, because if I try to draw now a 20 euro bill, it's not easy, actually, and we'll basically end soon. So I have not looked at a 20 euro bill for, I think, 2 weeks, I think, because now I pay almost everything with credit card, right? But I think I can try to make it, because I know that it's more or less, sorry? Network. Sorry. Not here? Okay. You're right. It's better, yeah. Sorry, I will make a mess, no? But at least I know that it's something like this, more or less. No? And I think that, for sure, on one side there is the map of Europe, more or less. More or less, something like this. More or less, okay? And, oh, of course, UK is there. Ireland, more or less, is like that, no? More or less. And then I think it has a 20 somewhere. And one side has windows, yeah, or bridges. I know that there is also a bridge somewhere. You see, I don't remember, but I think that this side has a bridge. Why? Because I remember that conceptually, when people designed the bills, they wanted to transmit the feeling that we are joining all Europeans together, blah, 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 okay? So there is a bridge, yeah? And I think there is a 20 somewhere, but I also remember that I think it's euro for sure somewhere in Latin and also in, I think in Greek, something like that. But you see, I'm not sure if there is 20 everywhere, or only, well, poor Ireland, or only, or only in the corners. I don't remember. It's amazing, but if I see the bill now, I will be able, right? So I have one. So we can compare. And this, oh no, this is not this one. Okay, so this is a 20. Oh man, okay, this was correct, right? So I have Europe on one side, it will vanish at some moment, no? Yeah, but take a look. What you see, I knew that were 20s somewhere, and you see the window here. So yeah, you can take a look. Yeah. But the point is that I made a few mistakes, but conceptually my brain encoded what I found was fascinating about this bill, right? The map of Europe, the bridge, Euro into languages, and a 20 somewhere. Yeah, and probably I made this mistake. Yeah, confused by the bills of the dollar. The dollar has the numbers in the corners, right? I think, correct? Okay, but the point is that, but you see, my idea is that we do not encode information like an image. We code it somehow, and it's very rare. I mean, maybe some other person will save a euro bill or whatever in a different format because there is something that, based on your education, you find more interesting. Okay, and yeah, and basically to warm up for Friday, I brought I brought one of my toys in the lab. So what we do in the lab is to try to achieve artificial intelligence, more or less, okay, or to develop ideas for artificial intelligence in which we use robots to fit the neuronal cultures, okay? I told you a few times, remember, that neuronal cultures lack any sensory input, okay? So that's why they are a bad model, or many people consider them a terrible model because you cannot reproduce the minimum ingredients of the brain, which is sensory input, okay? So come on, do it better, okay? And so our idea was, okay, at least let's try to fit the neural cultures with sensory input, okay? So in this robot, I have, these are ultrasounds, actually. We equip the robots with different sensors. This one has only the ultrasounds here, so you can detect objects based on the distance, okay? And actually like a bat, okay? And then we send information about the sensory input from the ultrasounds to the neuronal culture, and then neuronal culture interacts with this information and sends information to the wheels. Yeah? So this is our goal, to see if we can manage that the neuronal culture uses the robot as the body of the neuronal culture. Yeah, and the point is that if we can do that, if we manage to do that, then we can, in principle, train the neuronal culture to do particular tasks. And in principle, with just one watt or whatever, we can do some artificial intelligence, more or less, okay, or data classification in a much faster way than computers. Yes, sure.
Why with 1 watt?
Well,
Neuronal network, a lot of watts.
a lot of, no, but yeah, but I mean, the brain is 12 watt, more or less, yeah. So neural culture is much less, just by the number of neurons that you have there, more or less, just the order of magnitude, no? And just actually, and this guy, I mean, now we just program it, so it recognizes obstacles like that, okay, so I have to see. Okay, yeah, so tomorrow, oh, 1 minute, okay, we finish. Okay, but just on Friday, we will continue discussing a bit more with this, okay? And if you want more things, during the break or the afternoon, we can discuss more, yeah? Okay, thank you.
Okay, thank you very much. We can have only one question because we are very late. I mean, we have a tight day today. That's the problem. And
we can discuss in the break.
Only a 20 minutes break. Say 10 to 11, you have to be back here. Okay?
So,
That's because there is a colloquium in the afternoon.
so
An hour for
but we're going to stop here, or we can discuss maybe now in person during the break, maybe, no?
I mean, but we allow
Okay.
one question. So, okay.
Okay,
well,
thank you. Thank you. Where is my 20 euro bill?
The reason of all this tight schedule is because the talk in the afternoon will be at 2. So we have to leave early so you have time to eat.
Yeah, but the kind of network that you need for machine learning or AI to work have to be they have to develop this computer random. They need some perspective. Basically, you need a lot